Today, I'm going to give a lecture on how our histology slides are prepared. This is a topic on tissue preparation. Tissue preparation are steps required to produce high-quality sections or slices of specimens mounted on glass slides and appropriately stained to demonstrate whether the specimen is normal or abnormal. There are several steps in slide preparation. Tissue fixation is the first step in slide preparation that is needed to preserve tissue morphology and architecture and also to prevent cell digestion. There are two types of fixatives that can be used, formaldehyde or glutaraldehyde. The specimen should be fixed as close as possible to the time of removal of the organ from the body and should be small enough to be sure that the fixative can reach the most interior part of the specimen. The next step is dehydration, wherein the specimen is bathed in increasing ethanol to minimize tissue distortion brought about by diffusion currents. In this process, water is removed. In the process of clearing, alcohol is removed from the specimen by bathing it with a lipid solution like xylene or benzene. The alcohol is replaced with a solvent that is miscible with the embedding medium, paraffin. In this process, the tissue becomes transparent, hence the term clearing. The next step is embedding, wherein the specimen is now immersed in paraffin and baked to form a paraffin block. The tissue now is firm enough to be sectioned in thin slices using a microtome machine. Tissues to be studied under the light microscope are sliced 1 to 10 millimeter in thickness, while sections of tissues that will be used for electron microscopy are 60 to 130 nanometer or 0.06 to 0.13 of a millimeter. Staining is the last step. This is used to highlight and enable better visualization of the cell architecture. H&E is the most widely used stain in histology. So H&E, or hematoxylin and eosin, is the most common stain used. Hematoxylin is a basic dye reacting with the acidic component of the cells, such as the nucleic acid in the nucleus, staining these structures blue. The structures that readily bind with the basic dye, hematoxylin, are termed basophilic. Eosin is an acidic dye reacting to the basic proteins in the cytoplasm and collagen fibers, staining it pink. Since these structures readily bind to acid, it is said to be acidophilic or acid-liking. Although H&E staining is the mainstay of histological tissue examination, we need special stains to provide better visualization of specific structures based on their chemical, biological, and pathological characteristics. The first special stain is the periodic acid shift stain that reacts with glycogen granules, staining it magenta. This is the liver stained with the standard H&E stain with the basophilic nucleus and the eosinophilic cytoplasm. In a diabetic liver stained with PAS, the glycogen deposits stain magenta in color. Another special stain is ursine, which specifically reacts with elastic fibers, staining it brown to black. This is an aorta or elastic artery stained with H&E, whose main tissue component in the tunica media layer is elastin. Looking at it in higher magnification, the elastin is seen as pink wavy lines. When the aorta is stained with ursine, the elastic fibers react with a special dye and would now be clearly visible as black wavy structures. Silver is another special stain reacting with reticular fibers. This is again the liver using the H&E stain. When it uses the silver stain, the reticular fibers are now visible as black structures surrounding the hepatocytes. The reticular fibers are said to be argyrophilic or silver-loving. The right and Janssen stain are used to stain peripheral blood and bone marrow smears. This is an image of unstained blood cells. The next image would show the peripheral blood cells, mostly erythrocytes, a few platelets, and two neutrophils. In summary, I have discussed the importance of tissue preparation, the different steps that are utilized in tissue or slide preparation, and the different stains or special stains that can be used in order to visualize specific structures. Thank you.